Thank you so much for the introduction, and I'm really happy to be here. Nice talk, Dean, very good work. Nice to know you. I'm going to share my screen. Please let me know if this is visible to everybody. It's good.
Yes.
I'm kind of used to asking this to my students every single day, right? Can you see my screen? Can you see my screen? So I'm doing the same thing to here as well. Uh, really happy to be here. Happy New Year to everybody. I'm going to speak a little bit of a different language now, I guess. So please bear with me. If I have a very long presentation, please stop me. I'm happy to stop, and then I can culminate my talk where it has to be. The title of
Well,
my talk.
I
Yeah.
should I should chime in. I think I'm sorry we're sort of we're shortchanging your time a little bit, but there is a possibility for you to present a second part of it next week if you have time, because we can still have an open slot. But so
I see.
you don't need to rush if you want to split it in two.
I see. So I do have a beginning to the story, middle, and an end. So if I have to stop in the middle, I'll kind of, try and restart the beginning next time. All right. So the title of my talk is Merging Experimental and Computational Approaches to Study Neurodegenerative Disease Dynamics. This is something that I started when I came to UCLA back as a postdoc, and now after that, after I joined the Department of Integrative Biology and Physiology to both teach as well as to research, I kind of extended this project, and it's now become my research program. I started the Neural Dynamics group, which now has have many undergraduate students. We also have postdocs, and I remotely also have been mentoring graduate students as well. So in a very different sort of a context, I'm going to start off with human diseases. As we all know, there are many of them for whom the cause and these human diseases, they cause dynamic changes in cells, tissues, organism level, as well as even in populations. We are living in one of them, the pandemic. So they are very dynamic. And it is not really new to this group, where I can say computational methodologies and dynamic modeling are essential tools to decipher and even often simplify the complexity of these disease mechanisms, which happen at multiple spatiotemporal scales. Um, so especially in the type of disease that I study, these types of methodologies are not that much applied, at least at the resolution at which we want to understand the basic biology. So this kind of gives you an overview of the type of questions that we're asking and the type of approaches that we've been using. I call the title Microcircuit Vulnerability in Neurodegenerative Disease. If that's too much, I'll break it down as I go. Basically, we are looking at very simple neural circuits which are affected in diseases early on. And we are studying a specific neurodegenerative disease, which I'll talk about in the next slide. And we are interested in why certain types of neurons become vulnerable to cell death in these diseases at large. So the main question is, how does the brain, the puzzle of this disease affecting the brain chronically over a prolonged period of time as the animal ages, or as the human ages, I'm sorry, as the human as we age, and what are the underlying mechanisms? So I kind of, I'm taking an integrative hypothesis here where I put in lots of different things, and I'll explain that in a bit. But overall, we combine experimental work, computational modeling. Of course, we do use our models to conduct further simulations and apply theoretical analysis to better understand what is happening. And also, I'm really excited to share another methodology that we tried in collaboration with a lab at UCLA, and I'll tell you more about that. It involves interfacing real neurons with the computational models that we develop. So I'm really excited to share that with all of you. Um, So I thought this is something that's very relevant to this group. There are several computational approaches that we use, and we use them as complementary to the experiments. They just don't mean for us to analyze the data, but also in combination with the data that we collect, we want to drive a iterative process of models predicting and then the predictions being tested. And that, as we understand, that's really the goal of this whole modeling and the experiment connection. We try to do that at multiple levels. We use statistical approaches. We do data-driven modeling. Basically in systems which are coupled. In other words, connecting neurons to muscles and things like that. Mainly, the main thing I'll talk about today is dynamic modeling of cellular function. This is with the purpose of testing causal relationships between when we observe subcellular changes. How does that affect the emergent dynamics of these neurons? These are further taken into the network level, using dynamic modeling. And what we want to do there is to test the consequences on the patterns that the network generates, which can be predictive of the disease dysfunctions. Again, here, like I said before, I'm going to tell you a little bit about the real-time interface between the models and the neurons. And lastly, we are also using certain information theoretical approaches to in a very innovative way, I should say, mainly because these are things that have not been used to the type of mundane data, like the image data and things like that. I won't have time to cover too much of that, but I just wanted to list that here as we are using those. So one of the challenges with neurodegenerative diseases, it's a very long word, sorry, it particularly is as opposed to many other organ systems in the brain, after birth, neurons don't typically regenerate. So that's a big problem. And they form these complex interconnected recurrent networks, which makes it very difficult to sort of guess what is going to happen if you treated it in a certain way. Also, the brain is metabolically very demanding, and the nervous tissue is made up of very diverse cell types, which we don't completely understand yet. So their identities, their interactions are tentative. That makes it harder to sort of, apply therapies in a meaningful way. Also, the plasticity of the brain results in system-wide changes at the molecular level, structural level, as well as functional level. So that is another thing that is very difficult to predict and take into account as these therapies are designed. Again, these are just few things. There are many more. I just wanted to list those salient ones that came to my mind. And also, there's a lack of objective biomarkers for early detection of cell death process itself. So all of these, it makes it really hard to tackle these diseases and really have any type of cure for them. So now I'll introduce a little bit about the disease that I have been studying. Again, we do basic science, and I'm kind of trying to give an introduction in the context of the human disease and its etiology and things like that. This is called amyotrophic lateral sclerosis, or ALS. Some of you may have heard about this. This is also commonly called Lou Gehrig's disease in the US, based on this exemplary patient who got this disease very early in his life. Here are some facts and figures. This is rather a rare disease compared to Alzheimer's or Parkinson's disease. But then again, it makes it important to study it because the fatality is absolute. Patients die very quickly once they are clinically diagnosed of this disease. And also, it is a late onset disease like most of the other neurodegenerative diseases, but the causes are more or less unknown. So there are some known genetic causes, but mostly 90% of the time, the cause is rather unclear. And it's a very dramatically, it affects the neuromuscular system, it causing dramatic paralysis to the patient, which makes it really difficult to lead, even close to normal life. They suffer muscle, atrophy, and many things that lead to a devastating state in their lives. And it causes a lot of pressure on the caregivers. And again, there is no cure to this disease. So we wanted to understand what is the underlying pathophysiology, both from an experimental standpoint and also the dynamics standpoint. At the very basic level, what does this disease do? Well, it is known to affect a class of neurons called motor neurons. Motor neurons are my favorite, and they're very important because they basically link the brain, or everything that happens in the brain and the spinal cord, to the outside environment. In other words, they connect to the peripheral organs, the muscles, and therefore they are the final common pathway integrating all neural inputs, prior to that, and they make us perform the tasks that we perform, the movement that we show. So they're very important neurons, and they are present throughout the brain and the spinal cord. They're distributed all over, the nervous system. And in ALS, they are the ones that are the primary targets of this disease. So basically, motor neurons are known to degenerate, causing a retraction from the muscle, and the muscles losing that neural input, they atrophy, and that leads to paralysis, and eventually the patient dies, mostly because of the respiratory failure. But more importantly, their lives are extremely, devastating because of the nature of this disease. So, as we know, there are animal models of the certain diseases that enable us to do more invasive work, try and cut open the brain and see what kind of processes are going on in the disease, and how at the molecular level, cellular level, networks, and the organism level, how these diseases are affecting the system. So there are mouse models that are available for studying this disease, and they have advanced our understanding of the underlying pathophysiology to a great extent. And that's so one of the primary things that I wanted to share here is a image showing how this disease basically affects the neuromuscular junctions. On the left here, let me put on my laser pointer. So this is just in a normal muscle innervation situation where you have the axons innervating the muscle fibers here. These are the axonal terminals. And then here, this is a diseased mouse. And you see that clearly there is a lack of all of these terminals that are present here. And what is also shown in the figure down below here is that the axonal retraction or loss of these axons happens much sooner than the loss of the actual neuromuscular junctions. So in other words, the neural loss is happening prior to the actual muscle features of the disease that is seen. Therefore, this was a clear evidence that neurons are degenerating first, leading to the disease. So in addition to that, there is a lot of complexity in the type of motor system that we're looking at. For example, there are different types of motor neurons. Some of them innervate fast muscles, the others innervate slow muscles. So together, when they form these so-called motor units, they are important for normal function. And in ALS, as well as in aging, it is known that the fast motor neurons die first. So there is a progression relating to the type of the motor neuron that die first, or what we call selective vulnerability. So this is seen in ALS disease models as shown here. So at the very beginning, it appears as though the muscles are fine. The FF type of motor units are fine. The innervation is fine. But as time progresses, as you can see down here, the loss of these fast motor units basically causes some kind of a re-innervation from the slow to the fast fibers. But that doesn't last long. But once those start to be lost, then we start to see the paralysis and so on. Okay, so this is from a review article just summarizing the various possible mechanisms that are happening in ALS. Again, these are all have been possible because of these animal models that we use to study this disease. So it is not just, as you can see, appreciate here, there are several things going on. That's really the take home of this slide. It's a busy slide, but the idea is, as the muscle strength is lost, as the motor neurons are lost, there are also many different cellular processes that are occurring that leading to the neurodegenerative state. But so much is unknown. One of the things that I want to also highlight is that not just the motor neurons, but also supporting cells, which are the glial cells in the nervous system, they are also having a lot of pathophysiology here. So what was interesting to us back then was the fact that some of the studies indicated that there is a really early change in the electrophysiological properties of these disease of these mice or in the motor neurons. So this right here on the y-axis here is the age of the animal. As you can see, the paralysis, the onset of paralysis, etc., happens at later stage, at the day 100 or later. But then again, the disturbance in the electrical properties are seen early itself. So we got interested in that phenomenon, and we started looking at the motor neurons that control the jaw movement. Primarily because there are different types of ALS. It's not a single type of symptom that happens, and that in of itself manifests as a difficulty in diagnosing this disease. But among these different types, the so-called bulbar onset, which involves the facial muscles, the oral muscles, etc., that subtype has a much faster progression. Therefore, the decline or the survival time is much lesser in those patients. So we sort of focused on this area of the brain, which is called the brainstem, which houses these motor neurons that are controlling the jaw muscles, and also in supporting these ideas in the mouse studies, in the transgenic mice mouse model of ALS. It has been shown that even before the symptom is onset, there are several problems with oral function, like the animal is not able to lick as well, or chew as well, and therefore it is thought that because of its inability to consume food in a normal way, it loses the weight, and it is a side effect of the dysfunctions of the oral region. So our biological question was, what are the early signatures of disease vulnerability? So maybe if we understand this disease early on, maybe we can identify a longer window of opportunity to intervene in this. So this kind of started with my postdoctoral work here at UCLA in Dr. Scott Chandler's lab in integrative biology and physiology. I, in fact, started with experimental work. We targeted these jaw motor neurons that are controlling the jaw muscles, and we were able to identify them in the brain using a certain marker. And I performed electrophysiology recordings from these motor neurons. And due to the lack of molecular identifiers or markers for these different subtypes of motor neurons that I mentioned before, I ended up choosing a statistical approach, the clustering approach, to segregate or identify the different subtypes of motor neurons. This is thought to be nearly 98% accurate in determining what type of motor neuron those are. So we use this approach again. Using this, one of the big results that became evident here was the fact that the fast motor neurons were indeed selectively changing very early in this disease. So notice that this is pre-symptomatic at a very early stage. P10 corresponds to the postnatal day 10, whereas the symptoms start to manifest dramatically at about P90, 90 days or more. And the mouse only lives about P130 days. But even at P10, we were able to notice that cellular changes are occurring, especially, and they are selectively occurring in those motor neurons that are thought to be selectively vulnerable to degeneration. So we thought this was a very a strong case of electrophysiological signature for the disease itself. So I was then curious, since we studied a certain class of motor neurons, I was curious to see how that would manifest into a muscle dysfunction. So I took these data sets that I had collected and generated a muscle force generator model, which coupled the aspects of the motor neuron properties that we had observed into the muscle force generation. And this is a prediction that came out of that. So within a pool of motor neurons, as they start to be recruited to generate muscle force, which is on the y-axis here, you can see that there is a difference in the way that the force is generated in what I call a normal versus ALS condition. We interpreted this as a change in the dynamic range of the muscle, where in ALS, it appears as though very early, there is a really steep rise in this muscle force generation. So that we thought, of course, this requires experimental validation yet, but that we thought was an important finding and should motivate experimental measurements of these quantities. So after that, when I joined as am I too close to finish time?
I think you can have a few more minutes.
Okay, all right. So this is really, I should say, the beginning of my work as an assistant professor here at UCLA. So I got interested in how the motor neurons, which are dying, how is their function getting altered? So we were interested in reflexes that are very common. As you all know, we need our reflexes to be intact, to be functioning properly. If our reflexes suffer, we'll make many mistakes, and that will lead to, basically our, depending on what task we are performing, it could be dramatically a big problem. So in ALS, it has been known that in the human case, there are early polyphasic unstable fasciculation potentials in patients, and this reflects a reflex abnormality. So basically, your stretch reflexes are suffering in this case. It involves the reception at the peripheral muscle end, where you have the stretch receptors in the muscle, and that information is carried into the nervous system through these sensory neurons. And these sensory neurons, they relay that information to the brain, spinal cord, wherever else it has to go. But particularly, they activate the motor neurons that are right in that area. So the motor neurons immediately contract the muscle, and that's our reflex. So this is what we call a monosynaptic reflex. It's a one-stop connection. So these types of recordings are indicative of problems with these reflexes. So we started to look at, in the jaw circuitry, how is this reflex circuit getting affected in ALS? So that was our first stop. In order to do that, we picked these, again, we studied the jaw motor neurons, and interestingly, the circuitry, the sensory and the motor circuitry of the jaw system is very nicely placed, such that the stretch sensory neurons, which are the proprioceptive neurons, are placed inside the brain. So this made it possible for us to take a slice of the brain and have both the sensory and the motor in the same slice and actually study them. So we followed the same electrophysiological approach here. We looked at the properties of these sensory neurons. This work was mainly conducted by my postdoc, Dr. Soju Seiki, along with collaboration between Dr. Igor Spiegelman lab as well as Dr. Scott Chandler's lab. So here we found that these so-called proprioceptive sensory neurons were showing really disrupted firing patterns. So this is what we call, arrhythmia here, because as you can appreciate, the blue trace here reflects the normal or the wild type neurons, whereas the red ones are the afflicted neurons in the transgenic mouse. So clearly there was a lot of arrhythmic discharge going on in the action potential firing. For those of you who are wondering what these are action potentials or electrical signals in the neurons that we are recording from. And this is just a Poincare plot of the interspike interval. These are spikes, and the time distance between these spikes are interspike intervals. And we are looking at two consecutive ones, the one at a certain time and its previous one here. So this is a Poincare plot. It's kind of highlighting the idea that in the wild type, these ISIs are distributed in a certain way, but in the transgenic mouse, it is all over the place. So it's really disturbed. So that was a key finding here. And I guess I will complete this study and then pause, and I can take that from there. So we were then interested in what is causing this sensory abnormality, and Soju looked at another way to basically measure some of the ion channel currents that are causing these action potentials. So one key one in these neurons is a sodium current. These are mediated by a certain type of channels, which are called the NAV 1.6 sodium channels. These are really interesting channels. They are present in many types of neurons, and they have these very distinct characteristic that they have a fast component kinetically, they have a resurgent component, and then a persistent component. Now, the reason why I'm mentioning about all of these things is that it becomes important because we can associate one or the other component to be a way by which we can fix the arrhythmic firing that we saw. So in his study, Soju noted that these sodium currents were decreased in the transgenic mouse. And this is an undergraduate student, Kiera Quinn, who stepped in, and she looked at the protein expression for this NAV 1.6, which was consistently decreased in these neurons. So we had both functional evidence as well as molecular evidence there. So moving forward, so just let me know when I have to pause. I do have a significant amount left. So
yeah, I think when I think there are people who are moving to other meetings.
Okay, so all right, so to wrap this up, sorry, I didn't think that this would be a longer presentation.
Well, so, yeah, I think if you're able to wrap up a piece, and then because it's really interesting, and then maybe we can resume.
Sure. So in that case, what I'll do is I will pause here where we have seen sensory dysfunction, and then going further forward, I kind of want to take you through all of this, which is really interesting to me. But in summary, what happened based on all of our various efforts here was that we took the idea of this disease as a motor neuron disease, and we gave it this new way of thinking, which is a circuit-specific disease. So it's not only involves motor neurons dysfunctions, but also the sensory dysfunction that we have observed. And that then led to reflex dysfunction, which was predicted by a model that I couldn't show you, but I can come back to that in a different talk. Overall, it looks like there are changes to the function and the dynamics. I also had something to tell you about the neural information processing, and we also saw molecular changes as well. Again, there are several questions that remain unanswered, and I can discuss that a little bit later. Overall, I wanted to thank all of my collaborators at UCLA as well as at Ohio State. Primarily, this work was conducted in Dr. Scott Chandler's lab. Also, I'm really thankful to Dr. Ricardo Olchisi, Xia Yang, and Igor Spiegelman, Martina Vido. All of these investigators, they've helped us in various different ways to make these work possible. This was the most of this work, experimental work, was conducted by Soju Seiki, computational work by myself, there was another postdoc in Dr. Igor Spiegelman's lab who helped us with certain recordings. Many undergraduate students have been involved in this project. I can't name all of them, but these are the key ones. And I want to also thank Dr. Alan Garfinkel in our department. We had several productive discussions on modeling with him. The main support for this work came from the NINDS grant that I had, and various other things are listed here. Thank you all. I hope I could share this a little bit in more detail next time.
I would love to. Yeah, I think one of the questions that arises, like, how do in characterizing these neurons, the data that you get, there's imaging data and there's electrophysiological responses, and so it seems.
So what I showed you was all electrophysiology for now.
Okay.
The imaging data, we don't collect imaging data that is dynamic, in the sense that some people record calcium and other types of molecules, but we have mostly done voltage recordings or current recordings, which are limited to electrophysiology.
Yeah, and so it's those recordings that you then use for clustering, for identifying neurons using clustering approaches.
Right.
That would be nice to learn a bit more about, how these time series are actually clustered and what.
I see. Yeah. So I can talk to you at length about that. We kind of derive certain properties based on those recordings, and we use those as as measures to do these clustering. So
that would be really interesting to find out.
Yeah. But I do have some really cool recordings to show you where we actually drove the neuron using a model current that was sitting in a computer. And what happens is the model current updates the neuron's voltage and its firing behavior, and that feeds back to the model as the voltage, and the model updates based on that voltage. So it's a real-time closed loop control, and it's really remarkable. So very excited for that.
Very cool. Great.
Well, all right, thank you. Sorry, it was broken up.
Well, we got an introduction of the field. That's important.
Yes. I'm happy to give you a little bit more depth, in another talk. So.
Yeah, on the data types and the analysis. That's really what's, where we can intersect.
Absolutely.